Hello, I'm Brad Larson, former applications and field scientist and current product manager at Agilent Technologies. In this virtual demo video, I'm going to be talking to you about Agilent BioTek's Gen5 microplate and imaging software. Gen5 is a powerful and versatile software package that drives data and image acquisition, processing, and analysis for BioTek's readers and imagers. To help illustrate the power and flexibility of Gen5, when we think of how research proceeds, we can envision an inverted pyramid. You start broad and work towards more specific and detailed answers. Reader-based detection gives a wide variety of information quickly through the incorporation of an absorbance, fluorescence, or luminescence plate read, and can be used to gain mechanistic insight. Transmitted light and wide field microscopy let us dig a bit deeper and get more specific details, such as how many objects or cells play a role in the results we've seen. What are they doing and why? And how do they interact and change the environment around them? Finally, more advanced microscopy, such as upright or confocal imaging, takes us another layer deeper, particularly as many labs move towards more physiologically relevant model systems. These models could include scaffolds, spheroids, whole organisms such as caterpillars, C. elegans, and zebrafish, plant models such as Arabidopsis, or other unique designs. The fact that you can incorporate a single software package to help answer each of these questions from broad to focused is a huge benefit when you consider the time and effort that goes into mastering instrumentation software. For users new to Gen5, predefined protocols can be started with the push of a button, or quickly and easily created using the simple feature within the software. If a more experienced user wishes to build a protocol, the software also allows a great deal of customization to meet your needs. To optimize image capture, one can choose up to 6 imaging channels per step, in addition to image-based or laser-based autofocus. Capturing more than one image within a sample is also possible through the inclusion of montages, Z-stacks, and beacons. Or, if experiments are to be run over time, Gen5 can also capture images kinetically over seconds, minutes, hours, or days. Once images are captured, they can then be processed to improve downstream analysis. This can include background signal removal, deconvolution, stitching of image montages, projection of Z-stacked images, or even rendering of a 3-dimensional model. The flexibility of Gen5 also shines when you're ready to perform cellular analysis. A basic cell count and common metrics can be easily generated with default settings. But this is only the beginning. For example, as mentioned, an initial count will tell you how many objects are involved in the results seen from a well, or how many cells respond to a treatment compared to the total. However, if we then add a secondary analysis, we can also monitor what's taking place in other areas of the cell or organism around the primary mask by analyzing other metrics or creating your own. This is particularly useful when monitoring things such as mitochondrial activity, apoptotic activity within a spheroid, protein translocation, or other events. Subpopulations can also be generated via visualization or via statistical analysis through the use of scatter plots or histograms. Once you've generated quantitative data from your images or plate read, you can generate experiment-level readouts. For example, creating a dose-response curve based on gamma H2AX foci, or normalizing your luminescence plate read to cell count. For an even greater assessment of experimental images, Gen5 offers 3 advanced analysis modules and a software overlay. The first module, spot counting, allows individual objects to be counted within a primary or secondary mask that are specific to that object, such as counting the number of nuclei or lipid spots within a spheroid. Multiple spot analyses can also be made within the same analysis procedure for normalization purposes. The second module, Automatic Region of Interest, or Auto ROI, allows a large area of a slide or other vessel to be imaged at low magnification. Regions of interest are then automatically identified, and high-resolution imaging of the identified areas proceeds. The entire process saves both time and image storage space as imaging of unwanted areas is eliminated. The third module, Single Object Tracking, allows individual objects to be tracked over time to assess movement. Metrics such as total distance, Euclidean distance, and object velocity can be tracked during the entire time that images are captured. This is of particular interest for applications such as cancer-related chemotactic movement. The Neurite Outgrowth module supports neuronal research using single and multi-channel fluorescent images, multi-channel imaging with both transmitted light and fluorescent images, and single-channel transmitted light, typically phase contrast. The module automatically calculates soma and neurite metrics that are critical to outgrowth studies. And finally, the Scratch assay application enables 2D Scratch wound healing assays to be easily performed in either 24 or 96-well format. The app guides you through setup of the kinetic experiment to ensure images are automatically captured for the cell model being tested. Once this process is concluded, the app once again guides you through optimization of analysis parameters. Metrics such as average wound width, percent wound confluence, and max wound healing rate are then returned to assess treatments applied to test wells. Once images are captured and analysis is complete, data and high-resolution images can be easily exported out of Gen5. For data, anytime an Excel icon is seen, it can be clicked, and the currently viewed data will be copied into either a new or existing worksheet based on your preference. Single high-resolution images can also be saved in a variety of formats whenever the image is being viewed. Or, for bulk data and image export, custom power exports can be created as a template. Therefore, larger data sets and high-resolution images or movies will be exported automatically once the experiment is complete. Gen5 provides a wide breadth of methods to capture both PMT-based data and images, in addition to powerful analysis capabilities, thereby enabling a multitude of different life science experimental procedures to be performed without the need to incorporate other software. We hope that you have found this virtual demo video informative. If you have further questions regarding the capabilities of the software for microplate reading or imaging, please consult your local sales representative or field application scientist. Thank you for watching.